Over the past few years, in the area of travel medicine and infectious disease control and GI health, we've realized that when travelers like you and I go to Mexico or Africa or somewhere, we have the potential to be colonized with multi-drug-resistant organisms, and there's a certain type, extended spectrum beta-lactamase producing Enterobacteriaceae that are acquired. And more and more studies are showing that there's certain risk factors for going to a region like India or Southeast Asia, risk factors of just getting diarrhea when you travel, and then certainly the highest risk factor is if you have any antibiotics. And commonly when you get traveler's diarrhea, or before you travel, your doctor will provide you with a antibiotic to take if you get sick while you're traveling. So there's a lot of diarrhea and use of antibiotics in the travel setting. And they work really well in reducing your disease and ability to have your full itinerary. But they come with consequences, and one of the consequences we're understanding is this emergence of acquisition. So the impetus was trying to understand, well, one, are there any microbiome effects that suggest that someone is at higher risk, and what are the disturbances that might be associated with that? Our impetus for this particular study was because we just completed a large randomized controlled trial where we looked at single dose antibiotics, so just one pill of antibiotics, versus the standard of care, which is multi-day, multi-dose. We thought that there would be less dysbiotic or less acquisition, less disturbance, because we're just giving a single dose versus a multi-day, multi-dose. And so this was a study that was taking advantage of a large clinical trial that was done globally in my former job as a military Navy physician. So the key findings that we found was we looked at their subjects' stool functional microbiome before they got their treatment. We looked at it 7 days after, and then we looked at it at 21 days. And we didn't see a lot of change in the functional microbiome of those that were treated. There were some acute changes, but then they kind of reverted around day 21. We didn't see, we also looked for measures of antibiotic resistance. So we looked at a resistome, so like a compilation of all resistance genes in the stool that you can find in the stool, as well as some targeted genes that were specific to ESBL, producing Enterobacteriaceae. And we didn't see any real increase of those as well. And so our main conclusions were that the study was small, but that we don't see major disturbances and major resistance acquisition with single-dose antibiotics. We didn't have a comparison group, of a 3-day, multi-dose regimen. And we didn't have a placebo group. So our interpretation of our results have to be with caution. But the amount of ESBL that we detected in this trial was much less than that which has been seen in these observational studies. So if you go to Africa, on average, 35% of travelers returning will will have ESBL producing E. coli acquisition. We saw around 9%. So we saw, just overall, we saw less. But because we didn't see a lot of resistance acquisition, we couldn't really detect differences in the microbiome that would explain that. I think the important thing is we need to understand two areas. One is, what is the true impact of these infections on the individual travel, the average individual healthy traveler? How long do they hold it for? What are the health consequences of that? We also need to say we understand how the traveler fits in the bigger picture of local community emergence and sustainment of antimicrobial resistance and global spread. There's lots of sources of antimicrobial resistance spread. The traveler can play a role in that, but we don't fully understand. And so we need to do studies which help us balance or understand the balance between helping a traveler out who gets sick when they're on vacation or on a mission and the resistance spread globally that this acquisition might have. So I think that's the big picture of what needs to go next. I think what we're interested in doing is designing a clinical study that really might understand this, and so looking at placebo, single dose regimens versus multi-day dose regimens of the same drug in the same place, and look to see, we believe strongly that there's a dose effect, and so if you can minimize the dose of antibiotics that you use while you travel and get the same therapeutic effect, of stopping your traveler's diarrhea, then we should be doing that. And so we need more evidence to say that single dose antibiotics work, which we have that evidence, but it doesn't induce this acquisition that we're seeing with these multi-day dose regimens. And so those are the next areas of research that we'd like to pursue. I think that another area would be, what's interesting is people that acquire these multi-drug resistance organisms, we need to be able to screen them and then also get rid of it. And so I think there needs to be more emphasis and research in looking at decolonization, and that would be very helpful among not only travelers returning but also hospitalized patients that may be colonizing MDR, and reduce the major problem of antimicrobial resistance that we're facing.